I wanted to share with you some work I've been doing over the last couple of months, really my whole PhD, but formalizing it into something new the last couple of months. And that is basically being able to access and use knowledge graphs. And the way I'm doing that is now implemented in a new R package called KGExplorer. And I'll go into a little bit about what graphs are, what knowledge graphs are. And so, a graph more generally is a set of nodes connected by edges. And in the context of a knowledge graph, it's basically a way of formalizing knowledge that we have. And that could be within some particular domain, let's say genomics, where we have concepts represented as nodes, and then relationships between those concepts as the edges, the connections between them. So this could really, I'm describing it in very abstract terms because it really can be anything, but in the example here, we have a genetic variant that is associated with a phenotype, and that phenotype is also part of the disease. But these really can be anything. They, can go one direction, two directions, and they can have, different ways of connecting, could be associated with, could be causes, like a variant causes a phenotype if that's known. And on the left there, we have a very specific case of a knowledge graph, which is an ontology, where all of the nodes tend to be of one type, where, in this case, in the human phenotype ontology, they're all different phenotypes, and they are constructed hierarchically, but fundamentally that is still just a graph, where you start with terms that are very general and then move your way down to more specific terms. And there can even be some connections going across different branches. If, say, there's an immune component to some motor disorder or something like that. So that's the basic idea. And, I think this can be basically summed up as, knowledge graphs are graphs that attempt to summarize our knowledge in a particular domain. And, all the examples that I'll be giving here today are focused on disease biology. So in the example that I gave before, you can start by just a simple example of variant phenotype disease, but then, we would want to know some additional information about, well, what gene is actually mediating that relationship between the variant and the phenotype. And so you have this other connection here where, the variant is known to, regulate a gene through, being an enhancer or something like that. That's some piece of information that it's previously published, it's validated, and so we can draw that connection. And as a consequence, we can also start making indirect connections between that gene and the phenotype, and even the gene and the disease. And you can take that a step further and start connecting additional genes. So, we can start growing our network out by saying, well, this variant connects to this gene, and we know that this gene is very tightly co-expressed with this other gene. And you can grow your gene network out and say, well, there's some probability that both of these genes are actually involved in the phenotype and actually cause the disease. And in actually establishing those links and making sure that they're causal and not just associative, that's a whole lot of work, but in general, this is the kind of approach that you could take. And, this whole concept is very foundational to fields that we sometimes call systems biology or network biology. But really, even if we're not always thinking about things in terms of graphs or knowledge graphs, this is fundamentally what we do in a lot of science, and especially computational science. We are trying to make connections between different parts of biology. It might be, a cell type and a disease, or a disease and some other disease, mediated through genetic variants or something like that. Could really be anything. So you can take that a step further as well with beyond just having a yes/no binary connection. You can start adding weights to these things. So, in addition to the fact that there is a connection between the variant and the phenotype, the link is that it's been associated with it, as opposed to maybe being causal for it. And we can assign that a weight of, let's say, 0.22, which comes from a, let's say in this case, it comes from a GWAS, and these are normalized p-values scaled to a probability of 0 to 1. And you can really take any kind of numeric information, whether it could be discrete integers where it's, a score of 1 to 8, or it could be continuous like p-values, and you can start having strengths of associations, strengths of connections between these different nodes. You can also, in the case of variant to gene, you might want to take, some co-localization studies where, or even more simply, just taking a QTL and saying, well, in this tissue, we know this variant seems to be strongly associated with the regulation of this particular gene, and so we can say that that gene is regulated by this variant. And then you can take that step further and try to incorporate more information about the intersection between a variant, a gene, and a disease with something like GWAS QTL co-localization analysis. Like I mentioned before, you can also integrate gene-gene interactions through co-expression, or different databases like String, which include whole sets of known protein-protein interactions where they have physical interactions or functional interactions that are well characterized. And then you can also have a phenotype to disease, which might come from epidemiological data, or clinical diagnostic textbooks where we know that this phenotype is associated with the disease, and the frequency at which it's associated might be the connection weight between them. So, there are, motor symptoms, some like bradykinesia is associated with Parkinson's disease in, say, like 88% of the time, 88% of cases. And so we can draw that connection, whereas other phenotypes might happen less frequently, and that would be a weaker weight. So you can really start thinking about everything in this way, if you take it to the extreme. And it can be very helpful to think about things in this way, and to have a formalized way of trying to link information. And a lot of what we do when we're trying to gather data sets and then merge them together is connecting those nodes in the graph, running tests to be able to draw the links between them with some degree of certainty, or some kind of p-value. And as you start integrating more and more resources, more different data types, you can start seeing how things are, to some degree, or, either very closely or distantly, a lot of things are connected, especially in biology. As an example, our rare disease project is very much, in many ways, a graph problem, in that, we have information from some of these existing knowledge graphs, or what can be thought of as knowledge graphs, where we have phenotypes and we have the genes, and we also have information about how those genes are connected to phenotypes via different diseases. And we want to fill in some additional nodes here in the middle about cell types. And so we run a bunch of analyses to do enrichment of these gene sets to connect them to the different phenotypes and symptoms as like subsets of these genes that are mediated that are associated with a disease via a particular phenotype. But to get to that point, it's a lot of work. We typically, go through and just Google what is the best way to get each of these pieces of information. Then we have to basically learn the database specific format. Even if it's a table, we have to figure out what the column names mean, and then find ways of integrating these pieces of information together, which can be quite a challenge. And getting it all into one place for an analysis takes a lot of time. And that's traditionally how I've approached it. But there are some initiatives like Monarch, which are focused on disease biology and in particular genetics of disease biology, that attempt to link together many of these resources into one place and have harmonized data formats and even single merged files. So, in the extreme case, there is a single knowledge graph that Monarch distributes, which contains lots of different information. So variants, phenotypes, diseases, cells, different kinds of clinical courses, gene frequency, what else? Pathways, basically all the things that we typically are working with in the context of a lot of our different projects. And this amounts to is in a filtered down version of it without the variance, because that explodes the size quite a bit. You have over 525,000 nodes and 11.5 million edges. So it's a huge, huge network. And that's great that they have this all in one place, but it is, first of all, it's extremely difficult to find. I've been in this field for it's like 10 years, and I had no idea this existed. And I, only found out just, by total chance. And two, how do we actually start working with it? As it stands, it's a gigantic 3 gig file. With the variants, it would probably be many, many, many more gigs. And it's not obvious how you manipulate or search or work with this file effectively. So, I wanted to start making better use of this. So I ended up making I started writing functions into a couple of our other existing packages like KGExplorer for working with ontologies, specifically kind of thinking of them as a version of knowledge graphs, but then also multi-UC for pulling information about therapeutic targets, animal models, for different diseases and, perturbing different genes. But it stopped making sense to basically have these being put into these two packages, because it was getting so far from the original intent of these packages, and they were each kind of exploding in what they were covering. So I basically took the route of extracting all of the functions that I felt were very general to gathering all these types of information, and put it into its own package called KGExplorer. And it has a lot of functions for, getting ontologies, getting knowledge graphs, filtering knowledge graphs, visualizing them, and just doing a lot of the work for you so you don't have to worry about all the nitty-gritty of getting all this information organized. So what does that actually allow you to do? Like, why is that useful? Well, one of the first use cases that I started going into this for was we needed some validation for our rare diseases project. We've generated all of these associations between cell types and all of the phenotypes that have genes annotated them and from the human phenotype ontology. But these are all new associations for the most part, and we don't know how to validate them. So it would be really helpful to have some sort of ground truth, and this comes up time and time again with a lot of different projects in our lab. We generate predictions, we have these enrichment results, but we don't fundamentally know if they're right or not. And that's a very hard question. By using the knowledge graph from Monarch and building into this package, I was able to extract the nodes that were, any nodes that were connecting, that were a disease, a phenotype, or a cell type. And in particular, I was interested in diseases and phenotypes that were within the human phenotype ontology and its annotations that had known cell types. So these would be things like, some forms of myeloid cancer. They would be associated with myeloid cells, or glioblastoma might be associated with glia or something like that. And there's certain examples of very well established. Many of them it's in the name of the disease or the phenotype. Very well established cell types that are known to cause these diseases. And what you end up with after doing all of that filtering for this much larger graph is all of these nodes and connections. And it's formatted in this R object called a table graph, which is a pretty efficient way of representing this information, because you basically have your nodes and all the metadata for each of those nodes. So that includes the diseases, the phenotypes, the cells. And then you have the edges, which are the connections between them, and some description about, the information about where those connections came from, the strength of the connections, publications that support those connections. And everything has a unique identifier, both the nodes and the connections, so that it makes it quite easy to be able to search through this. So, in this case, I basically pulled in all of that information. You have the cell type ameloblasts being associated with the disease term ameloblastic carcinoma. And you'll notice that it has mondo terms, which are all human diseases that are connected to it, but it also has mouse phenotypes from the mammalian phenotype ontology. So it's not limited to just human. You can start looking at other species as well, pull in that information. But if you want, you can filter it down to just specific databases that you want connections with. And so we were able to basically use this and say, we want our, when you talk about connections between nodes, you can describe it as a subject and an object, or you can just think of it as from one node to another node, and there's some connection. In this case, I filtered it down so that the object was the cell type and the subject was a disease or a phenotype. And for this project, we filtered down the subject to just phenotypes that were within the human phenotype ontology. and then get all of those cell type phenotype associations so that we can say, you know, in our enrichment results, what percentage of these known cell type phenotype connections do we actually recapitulate and actually compute some recall metrics to give an idea of how accurate we are. One of the functions within KGExplorer is to has a lot of functions for being able to switch back and forth between different object types, and the main ones are those graph representation over to a simple data table, because I realize that not everyone is used to thinking in terms of graphs and table graphs, and it may just be helpful to have a simple layout, a table that you can open it up in Excel and just go through. So
you can just say, I want all the diseases that are associated with variants.
Yeah, you could do it that way. It really, that's the nice thing. It's anything connected to anything. And you can even do things to say like, well, I want diseases that are associated with phenotypes that are associated with variants. And so you can start specifying things like, you know, I want a disease that is connected to a variant with n degrees of separation. You know, it's mediated by 2 or less nodes in between. And there's all different ways you can draw connections between them. And that logic can be a little bit difficult to formalize. So part of what I'm doing in this package is to make that much simpler, have very flexible ways of filtering, directly on the node metadata, directly on the edge metadata, and then also about, you know, how things are connected to each other, so that, you know, for example, I want diseases, let's say I want phenotypes in mouse that connect to a gene. However, I also want to filter by phenotypes that connect to genes that connect to the human orthologs. So they need to have a human ortholog for me to care about it. Because I want to use it downstream for something where I'm comparing with some human gene list. So yeah, it can even be, it's not just, you know, connections between different categories, but it can also be within categories. In the case of genes, this amounts to a mapping from, you know, the mouse version of a gene to the human version of a gene to a zebrafish version of a gene. Those are all connected concepts. And a lot of the pieces of information you get out of this are mappings across different databases. So let's say I want to take the Mondo ID of Parkinson's disease, subtype 3A or whatever. And I want to find out, well, what other database IDs does that relate to? Like, what is the ID? What is the equivalent ID for that in the disease ontology, which is a separate ontology? Just getting that information is often shockingly difficult. And part of the goal of this is to make mappings across IDs across databases much easier, because so far I haven't found a unified way of doing this. It'd be nice if there was, but you know, if you're given a data set that has identifiers in one format and you need to merge it with a data set that has identifiers in a different format, you know, there's a lot of things that are comparable, but you don't want to have to manually go through several thousand, you know, combinations of identifiers to be able to create those mappings. You know, this information should already be known. There's a lot of those maps already exist. It's just a matter of accessing them and using them. Another use case is to extract known animal models that have a given phenotype that also exists in humans. So in this case, we're kind of conceptualizing the nodes as, let's say, starting with a human phenotype, and then how does that map onto the equivalent phenotype in a mouse or a zebrafish or a frog? And, what are the genes connecting, first of all, what is the similarity between those? Like, how similar is, thrombosis in a mouse compared to thrombosis in a human? There are some quantitative metrics for being able to say what is the overall similarity of those. So how well does the mouse version of that recapitulate the human version? And that's a connection between those two with some weight. But then you can also start, connecting things like, I want to take the human phenotype and all its human genes, the mouse phenotype and all its genes, and then link those mouse genes to the human orthologs, and then compute the overlap in the genes. That's another way to get similarity at the gene level. And so I wrote in some functions for doing this exact problem, where it pulls in all the relevant information, gets some existing similarity metrics just based on descriptions of phenotypes between the mouse and the human version, but then also computes another similarity metric based on the genetic similarity. And so you can merge those together and have what I'm calling a U-pheno equivalence score, where it's kind of the mix of how similar are the phenotypes and how similar are the genes underlying those phenotypes across the two species. And you can basically, for all on the y-axis here, we have a bunch of phenotypes that are in humans, but then also have homologs in these other species, including mouse, rat, Deniurario is zebrafish, C. elegans, and they're all sort of categorized. These phenotypes are categorized in different parts of the human phenotype ontology, and we can say, like, all right, for leukocytosis, is there an equivalent in rat? Well, there may be, but we don't have a mapping here. And there is a mapping between zebrafish and human with each of their versions of leukocytosis, but it's actually not as strong it's not as similar as the version between mouse and human. And so we can actually have some quantitative metrics to say, like, if we want to model a disease, or we want to model a particular gene mechanism within a disease phenotype, we can basically look to this and say, well, this species has the best shot of actually recapitulating, or there's actually some good evidence that says we will be able to model this in a rat or a mouse or whatever. Now, obviously, as I mentioned before, this is going to be limited by the information that's available. Some of these mappings are not as complete as others. Part of the reason you see mouse being much better than rat is, they're equally related to us, 90 million years of evolution, but the mouse has just been studied a lot more. So that is something to keep in mind, but nevertheless, it's a very systematic, high throughput way of being able to assess whether there's a model out there for your given phenotype. And you can even put these together and say, I want to model this disease, which is characterized by all these phenotypes. What is the likelihood this is actually going to translate? And then finally, as a last example, we can take parts of the knowledge graph that Monarch has put together that doesn't include the variants, and then merge that together with some other data that's currently separated. about the variants and their associations with diseases, phenotypes, and genes. And so, I have this drawn as diseases to phenotypes to genes to variants, but it's really connections between all combinations of these things. And you can filter to what types of connections you want to keep. But the idea is, we want to focus on all these things, and maybe we want to pull out a subset of diseases and all of their connections with phenotypes, genes, variants. And as an example here, I pulled out this subgraph. I just kind of randomly sampled the millions of results that came back. And you can see here an EFO term, which is usually used in GWAS. Platelet crit is the phenotype that was being quantified here. And these are a bunch of the variants that have been associated with that, presumably through a GWAS. But then also, interestingly, you do have connections here between the human phenotype ontology term and different variants, but these variants are actually in mice. So I think if I were plotting the full graph, what we would actually see here is this human phenotype ontology term be connected to a mouse equivalent mouse phenotype, and then they're both connected to all of these mouse variants and also some human variants. And so you can get very, very detailed in terms of what kinds of relationships you want to look at. But of course, we're usually trying to answer a specific question, not necessarily doing this really high throughput version. So all of these functions that I've been showing in the upper left are meant to make that super simple and intuitive.
You can modify the data set and say, like, we've decided these phenotypes are related. You can draw lines onto the graph.
You can modify the data set in your R object. Yeah. You would need to go, in terms of updating the knowledge in the knowledge graph as it's distributed by Monarch.
But just within a session.
Yeah, oh yeah, within a session, absolutely. You can modify it however you want. You could even merge totally different graphs. Like I could take in, I could take in a subset of this knowledge graph that connects known phenotypes to cell types, and then merge that with our data from from the Rare Diseases Project, connecting phenotypes to cell types, and then, plot one big graph using that. Yeah, so you can manipulate it whatever way you want. I'm using a package called tidygraph to basically manipulate these graph objects. And there's a whole host of functions for being able to do those kinds of merges. So just to summarize, this is still very early days for KGExplorer. It's right now I'm just focused on getting it to work in the context of our project for the rare diseases, but I'd like to over time keep chipping away at it and adding features. I've been in contact with some of the Monarch team who are interested in building more efficient querying tools for the Monarch knowledge graph. They recently launched a queryable database in Neo.js format, which is basically just a very efficient format for querying graphs and doing very complex queries with that. So you don't have to read the entire thing at once. And then, of course, there's other databases beyond just Monarch. Open Targets has a whole host of data. ClinVar, Therapeutics Target Database, BioCipher. I have to think a little bit about exactly which ones are actually necessary to pull in. For example, ClinVar.
I guess most people here have no idea what any of them are.
Okay, sorry. Open Targets, very similar to Monarch in the sense that it's trying to aggregate lots of information from mostly genome-wide association studies, QTLs, put it all into one big database that's searchable, and being able to extract specific p-values of associations, then doing all these downstream analyses for linking, doing colocalization between GWAS and QTL, and just providing all of those results on their server. And they have an API as well as an interactive browser. ClinVar is a longstanding database created by NIH to basically have a huge database connecting variants, genetic variants to diseases. And in contrast with open targets and some of these GWAS-derived databases, ClinVar is primarily focused on rare variants that come into the clinic. Kids that come in, kids or other, adults that come into the clinic and they have some sort of rare disease, and they're through, genotyping or genome sequencing, they're able to ultimately identify a causal variant, and or some associated variant, and then they can differentiate those annotations in there. But yeah, it's many millions of variants, tens of millions of links between variants and diseases at this point. And I should mention that Monarch does ingest ClinVar as part of it, but there's some additional metadata in ClinVar that can be helpful in terms of what those variants are actually doing, whether they're
classification of variants.
Hmm?
Classification of variants also.
Yeah, exactly. And some of them, many of them, in fact, are variants of unknown significance. So we don't necessarily have that information, but they do have classification for a good number of them. So whether they're protein coding mutations, whether they're within an exon or they're intronic, and they also incorporate, if it's within a protein coding region, then it will actually incorporate predictions of pathogenicity, whether it's benign or likely to be pathogenic, because sometimes some people have a variant, it's very strongly associated with, that disease, but we don't know if it's actually causing it or how it's causing it. And then you also have, I've noticed a lot of these databases now are incorporating predictions from AI models like Splice AI for characterizing things that we just don't have enough experimental data to be able to verify. So Splice AI is basically a gene sequence to splicing function predictor AI model, which basically says for any point in the genome, if you mutate it, how likely is it that it's going to cause some sort of change in the splicing behavior of the gene? And it's the best in class. It was published in 2019 originally, and no one's been able to outcompete it yet. And now it's actually being integrated into a lot of clinical genomics pipelines used in screening in clinics. And so they have, all sorts of, all different ways of annotating these variants. And so you would want that additional information if you're, say, developing a gene therapy that is targeting a gene that is known to be associated with a phenotype, but then you want to know specifically what variants should you be targeting within that, and what mechanisms are you going for? Like, is it a translocation or some large structural variant, or is it something that might be amenable to CRISPR editing, where you can go in and actually just edit a single base pair and fix the disease that way? Therapeutics target database, it's a big database of both small molecules and gene and cell therapies, other biologics, basically any kind of therapy. and linking it to specific diseases. And it gives an up-to-date accounting of where all of those therapies are in the clinical trials pipeline. You know, whether they have been approved for years or whether they're still in preclinical testing or phase 1, 2, 2A, 2B, you know, it gets quite detailed. It's just a nice way of assessing where we're at in terms of therapies for specific indications. And then Biocipher is another kind of like big meta knowledge graph, like Monarch. But their innovation is that they're using large language models to help query and use the information and extract the information from it. So they're actually building in, you know, connecting the LLM to the knowledge graph so that it can, you know, it can give you information, but it's actually validating it by pulling out real data. So yeah, there's a lot of areas you can go with this, both from an engineering standpoint to make this easier and faster, but you can also start doing things like embedding concepts based on, you know, how similar are diseases to each other? Are there, you know, maybe there's a drug that might be helpful for treating a disease because of its similarity to another disease, all sorts of graph theory challenges that you can start addressing with this. You can use graph neural networks to try and do some of that, and you can also do some causal inference, which is a step beyond just making associative inferences, where you're actually trying to infer the causality between two nodes, or, you know, that are adjacent to each other or maybe indirectly associated with one another. So that's pretty much it. Does anyone have questions?
So why is it easier than just querying a table?
If the table has everything you want, then querying a table is probably easier. But usually there's not one table that has everything that we want. And it usually involves doing lots of joins between many different tables. And if that's what you're trying to do, it ultimately becomes a graph problem. Because otherwise the table gets wider and wider and wider.
But most of the time, you don't really want to go beyond like this table to that table. Right?
Depends.
Which is not that hard. You join two tables together, right? So
Yeah, for simple cases, it may not be an issue, but, you know, I think when you really dig down into it, there's usually more steps. So let's say I want to join table 1 to table 2, but the genes are in different species formats or something like that. And so we want to do the mapping over, or the disease IDs are not in the same format, so we need to map those over. It really depends. For very simple use cases, yeah, joining two tables is probably fine. But as the information that you're trying to collect gets more and more complex and you're pulling in different elements, then having a graph-based approach can be helpful. And it also means that, you know, maybe once you do get it to two tables with matching column names, it is easy, but to get to that point, you have to do a lot of digging and a lot of formatting to get that information so that you can do that merge. Whereas having a large knowledge graph with all that information already in it means you can just query one place and get all the information you need in the best case scenario. And then whatever you don't need, or whatever you don't have within that knowledge graph, you know, it makes it a bit easier to do complex merges and filtering with some other data set by pulling that in. Because let's say, you know, I need to merge by 3 different variables and 2 of them are mapped in the same way. There's a series of steps that it takes to get to that normally, and you can do that sort of linking more efficiently with a graph without having to know all of the nitty gritty in between.
And Monarch is actually taking care of updating everything. It's not like they did this once, grant finish,
No.
it's never going to be updated again.
Yeah, right. So this is a tricky part with knowledge graphs, because there's been plenty of knowledge graphs published over time, plenty of databases that claim to be the one to rule them all in biomedical sciences, and many of them have gone defunct after the initial publication. So being able to distinguish those is difficult. Monarch has been around for a while. It's part of the larger initiative that includes human phenotype ontology, Mondo, some of these other things that have been around for at least a decade. They seem to have pretty consistent funding and staff. They seem to be pretty organized, and you can see exactly when they make updates, both on their server and on their GitHub pages. And one of the other nice things about their graphs and all the tables they provide is that they are timestamped for when they were entered. So you can actually have an idea of not only when it was entered into the Monarch database, but when was that knowledge entered into the primary databases that it's ingesting from. So in the case of ClinVar, when was it deposited in a ClinVar first? Both those pieces of information are available. So it has both the primary and the secondary sources, which is important because the more data you pull in, the more version control becomes important, because there are big differences between HPO 2019 and HPO 2024. And same goes for a lot of these data sets. They're constantly growing. ClinVar has grown massively since one of its older publications, and that really does change. It can fundamentally change the properties of the data. Like, I think when ClinVar had a report published in 2016, there were hardly any known splicing mutations in there. And now that they have Splice AI and able to make predictions across the genome, they were able to go back and reclassify many of those variants of unknown significance as likely splicing mutations that are deep intronic, that are not classical, really obvious splicing splice sites.
So is it like every time before the user before they can find information, they have to update the Explorer with updated database? Right?
No, so because the data is not stored within KGExplorer itself, it's making a call to the external database, and that external database is continuously updated. Now, there are some features to try and speed things up, like when you're downloading large tables that are really, that take a while to download, it'll do some caching, but you always have the option to clear the cache and re-download a new version to make sure you're using the most up-to-date version.
So it's like every time when I use it, they will call the information from
Yeah.
elsewhere.
Yeah, exactly. Unless you within a session, you want to use the version you just downloaded 10 minutes ago. That if you're doing some things over and over again, that's helpful. But, if you return to something a year later, you probably want to use the updated version, and you have the option to do that as well. Yeah. But yeah, ultimately it's sourcing all of the data from the original data set, or as close to it as you can get. I guess with Monarch's knowledge graph, they are pulling in information, but yeah, this comes back to like, you know, do we trust them to be constantly pulling in ClinVar and pulling in, but they've done a lot of work with having pipelines that do this automatically. I think with one of the downfalls of a lot of these other knowledge graphs is they make a knowledge graph, but it's all manual. Or it's like one-time use scripts, and then when they break, no one ever updates it, and no one ever runs it again. Whereas Monarch actually has like a schedule to do these data pulls and has, you know, these ingestion adapters and pipelines to basically pull in the data on a semi-continuous basis.
Is it set up as a Neo4j database at the moment?
They
My
just,
understanding is most graph neural network software
yeah,
take Neo4j as it.
yeah, they just set up the Neo4j. Well, they have the files available. You can download them right now in Neo.js format. But does require a little setup to do some local hosting with that file. What they just started was doing the hosting on an external server, which you can then query directly without having to download the entire file and set it up locally and then querying that file. So it's just it'll be an easier way of doing it. And the collaborator at Monarch is trying to do that latter version with the R package, so that, you know, you don't have to have anything downloaded, you just query the data, you can only pull that in. So, yeah, a lot of data out there, and it's always a challenge figuring out, you know, what's a worthwhile investment in terms of figuring out how to use this data and cleaning this data and formatting it. And I'm always looking for ways to make better investments and get some longevity out of it, because there's been so many times where I, you know, download a data set and then, you know, it's defunct or it's a different format sometime later, and, you know, having some standardized way of accessing this information is really helpful.
Okay. I do think there is potential in the future for things like what we discussed with. So tomorrow we're meeting this lady in Southampton Uni. She's a geneticist. And so Brian's been using this human phenotype ontology data, trying to map that onto cell types. She's been taking that and trying to work out, can you use all the genetic information from across the UK to say whether an individual person will have a phenotype? Like, do they just have a burden of mutations within genes associated with a trait? But if you imagine at the moment, you've got all this data there, all the cell types, all the genes, all the phenotypes. By knowing what cell type causes a disease, we can probably make guesses about what genes probably cause disease, even if we don't actually know that they cause diseases at the moment. Like, if they're specific to this cell type, they're known to cause other diseases, they have variants in them. You can probably fill in the gaps. Like, graph neural networks have been found to be fairly good at doing that kind of thing.
Yeah, and yeah, the advantage of having something like a graph neural network is it fundamentally borrows information across the network.
Yeah.
It's kind of like it's filling in a lot of the gaps where there's a gene that we didn't know was associated with the disease because we just didn't have that link with the cell type or something like that. And they work well when you have many examples, and they tend to be more robust as you have more examples. So maybe that, you might have a case where a gene is important for mediating a disease effect in multiple cell types, and you might have multiple examples. And then you have some negative examples too, where it's not mediating it through other cell types. And there's some very complex logic and, being able to distinguish those scenarios, and graph neural network may be one way to basically making those links and saying like, well, we think it's associated with this, but not this cell type. Or we think it's associated with this gene or not this gene.